Hi everyone, I'm Calvin and I'm from the Godfrey Lab based within the Doherty Institute at the University of Melbourne, and I'm going to present highlights from my JM paper entitled Selective Regulation of Interferon-gamma and IL-4 Co-producing Unconventional T Cells by Purinergic Signaling. Unconventional T cells like MAIT, NKT, and gammadelta T cells are a family of T cells that recognize non-peptide antigens. These cells together can comprise up to 10 and even 50% of T cells in sites like the blood, mucosa, and liver. And these cells can rapidly produce a variety of cytokines upon activation. So specifically, T-bet positive unconventional T cells produce interferon gamma, whilst their raw gamma T expressing counterparts produce interleukin-17. While some gamma delta and NKT cells can simultaneously produce interferon gamma and IL-4, it is not clear how this is regulated and whether MAIT cells are even capable of this co-production. So via their potent cytokine responses, unconventional T cells can contribute to immune defense, though it can also mediate tissue damage. How these cells and their functional subsets are regulated by tissue damage is not clear. So damaged cells can release metabolites like NAD, which, via an enzyme known as ARTC2, can trigger the activation of a purinergic receptor, P2X7, and this can drive cell death. Whilst NKT1 cells are susceptible to this NAD-induced cell death, it is unknown how this affects MAIT and gamma-delta T cells, as well as unconventional T cells as a whole. So we found that T-bet, but not raw gamma T positive unconventional T cells co-express ARTC2 and P2X7, especially in the liver and the spleen. In response to cell damage ex vivo, we show that liver unconventional T cells undergo increased cell death upon cell culture, and this could be prevented by blocking P2X7 or ARTC2. And we also showed a rescue from cell death following antigen encounter. Furthermore, treating mice with NAD selectively depleted T-bet positive unconventional T cells, as depicted here, and this could be partially rescued via prior ARTC2 blockade. Analyzing cytokine production, we next show that blocking P2X7 or ARTC2 drastically increased the frequencies of unconventional T cells that produce both interferon gamma and IL-4, revealing the existence of interferon gamma and IL-4 co-producing MAIT cells. And interestingly, subsets of alpha-beta T cells that also produced both of these cytokines. So overall, we believe that upon tissue damage, the release of NAD triggers ARTC2-mediated P2X7 activation to deplete T-bet positive unconventional T cells. This likely reduces inappropriate cytokine secretion by these cells to prevent further inflammation and injury to our tissues. Our work thus shed light on how unconventional T cell functional subsets can be differentially controlled in the immune response. Thank you very much for listening.